Welcome to Engineered Gene Drives, Regulatory and Policy Considerations. This is a webinar series brought to you by the Gene Convene Global Collaborative. My name is Hector Kimata. Thank you for joining us today. I'll be hosting and moderating this series along with my colleague, David Obrochta. We have two speakers today, Dr. Michael Bonsall from Oxford University and Dr. Owen Edwards from CSIRO. But before they give their presentations, I have a brief introduction to Gene Convene. The Gene Convene Global Collaborative is an initiative within the Foundation for the National Institutes of Health that results from FNIH's longtime involvement with vector-borne disease. Now, we at Gene Convene see that there is the potential for genetic control, genetic biocontrol, and gene drive technology specifically to contribute to reducing and even eliminating vector-borne diseases like human malaria and other urgent public health challenges. But this potential still has considerable uncertainty. If we explore these technologies responsibly, though, we can evaluate whether they're safe and effective and can see if, in fact, these technologies can achieve what's been claimed for them. The Gene Convene Global Collaborative works to advance best practices and inform decision making for development of genetic biocontrol technologies to improve public health. And one of the ways we do this is to offer accurate information, advice, and technical support so that we can foster responsible approaches to research and governance of gene drive and other genetic biocontrol technologies for public health. And this webinar series is part of that effort. You can find out more about the Gene Convene Global Collaborative by visiting fnih.org/Gene Convene. Now, we've also created an online resource, the Gene Convene Virtual Institute, to share knowledge that we hope will increase the understanding of gene drive and other biocontrol technologies. In that resource, we collect, track, organize, and share things like journal articles, media reports, and policy and regulatory documents. And we hope this will be a useful resource to anyone interested in genetic biocontrol and gene drive. So please check out the Gene Convene Virtual Institute site. And while you're there, sign up for the weekly newsletter, which keeps you up to date on the latest developments in this area. And also, you can follow us on Twitter and Facebook. We're also still looking to hire a web assistant to help develop and maintain the Virtual Institute's digital presence. So please see that site also for future information or for further information. So now this webinar series, we're in the third session of this series of 5 that's focused on regulations, policy, and governance. The first week we had speakers on global regulatory frameworks, and last week we looked at the regional level. So this week, our two speakers will be talking about how other disciplines can inform risk assessments of gene-drive organisms. Last week, we heard about the areas where further development of risk assessment tools might be needed. So today, I anticipate we'll be hearing about the tools that are used to deal with other organisms that present risks similar to those posed by gene drives. Now, as always, I have some housekeeping items. For any technical problems, please contact FNIH Events at the email address you see here. They're monitoring their emails in case you have problems. The first hour of this session will consist of the two presentations, and at any time during these, if you have any questions, please write them down using the chat function of Zoom, and we'll address them after the presentations are done. You'll find the chat at the bottom of your screen in Zoom. The webinar session will end at exactly 90 minutes. If there are any questions left, we'll address them offline. This webinar is recorded and will be posted at the Gene Convene Virtual Institute site along with all questions and their answers. Now I'd like to introduce our two speakers. Mike Bonsall is professor of mathematical biology at the University of Oxford. He leads a research group that focuses on integrating theoretical and empirical problems in the life sciences. Particular themes of research in the group range from problems in mammalian developmental biology and stem cell dynamics through to ecology and evolutionary biology, GM insects, the dynamics of communicable disease, and non-communicable psychiatric disorders such as bipolar disorder and PTSD. He has worked at the interface of policy and science around GM insect releases, serving on the UK DEFRA Committee for Releases to the Environment, and also working with the House of Lords Science and Technology Committee, the European Food Safety Authority, and the WHO. Owen Edwards conducts research at CSIRO focused initially on the ecological and molecular basis of aphid host plant interactions, and then expanded to investigate molecular interactions of aphids with their environment more broadly, including epigenetic regulation of aphid polyphenism. Owen continues to serve on the board of the International Aphid Genomics Consortium and on advisory committees to many other invertebrate genomics consortia. Building on his expertise in invertebrate genomics, he leads a CSIRO research group in environmental genomics, which includes a research team focused on genetic pest control technologies. Most recently, Owen was appointed as leader of the environment and biocontrol domain of CSIRO's Future Science platform in synthetic biology. Within this platform, he oversees projects delivering environmental outcomes, including gene drive for biological control and engineering resistance or resilience in threatened ecosystems. So with that, we'll have Mike Bonsall go first, and I'd like to turn over the webinar now to him. Mike, are you ready to go?
Yep. Can you hear me, Hector?
Yes, I can. Yeah.
Great. Excellent. Great.
You can go ahead and share your screen.
I will. Hopefully you can see my screen, everybody.
I can indeed. Yep.
Excellent. Great.
Great.
Okay. Thanks very much for inviting me, Hector. Great pleasure to be here. As Hector said, we have over the last few years been involved with various agencies looking at regulatory approaches to gene-drive organisms. I'm not speaking on behalf of any of those organizations. I am here in my own capacity. So anything I say is not attributable to either any of those. So I'm really here to tell you about the work that we do in the group and how that sort of interfaces with the policy work that we've been involved with. I'm based in the zoology department in Oxford, and as said, lead the mathematical ecology research group. So what I'm going to do is give you sort of like a way in which we think about sort of risk frameworks and how we might think about using technologies that are already out there to help us inform about policy and regulatory frameworks for gene-drive organisms. So the talk I'm going to give you is going to start by giving you sort of like what we think of as a sort of pathway to harm and a risk hypothesis framework that was just going to broadly set the scene for thinking about these sorts of ideas that we have. And then I'm going to step through, because one of the things we find really hard, and one of the things we find really hard, and not saying anybody else does, the one thing I find really hard is how we actually define harm. So we're going to think about three ways in which we can think about harm. We're going to talk about, I'm going to give you the idea, like harm one, the idea of risk of adverse effects. And I'm going to introduce you to a little bit of science, a little bit of some of the modeling work that we do to help us how we understand those effects. And I'm going to talk about the sort of the policy and regulatory approaches that sit within, is this effect harmful? So that's going to draw on existing regulatory frameworks that are in place for other organisms or for other approaches to controlling, particularly insect pests or vectors of disease. And then I'm going to think about, is the harmful effect acceptable, and thinking about the implications for gene-drive organisms. So let's get into this a little bit. So when we're thinking about pathways to harm, we're really thinking about this is a sort of like this sort of classic archetypal sort of approach to thinking about how we would approach an environmental risk assessment of a modified insect. And we're really thinking about the impacts of this on human health and wider biodiversity in the environment. And we can sort of partition this. So the really crucial thing is really at the top is really thinking about how we identify a hazard. What is that? What is it that we're really concerned about that has this potential detrimental effect on the environment or human health? And it's really important that that is predicated on a risk-based approach rather than something that we might think of as a science-based approach, or something that we may just generically be interested in. There has to be a really good way, and I'll give you an example of that later in the talk about how we've formulated this sort of idea for these different technologies and for a hypothetical gene drive. And then what you want to do then is to sort of characterize under that hazard. Once you've identified that hazard, is characterizing that hazard and thinking about the sort of characterization of the exposure to that hazard. and then working through the risk evaluation of that. And it's really important. And I think one of the things, I may be talking to the converted here, that actually this idea of characterizing the hazard and characterizing exposure turns out to be a product. It's a multiplication to give us some value of some risk evaluation. And it's those two things. So it isn't just, what tends to happen is that we just go down the left-hand side of this, and we concentrate solely on the hazard without actually characterizing exposure and the risk or the consequences that has for calculating risk evaluation. And risk evaluation then falls into two things. We identify a risk and risk management, and then we think about how we mitigate that risk. And we really have to think through all of that in order to do our environmental risk assessment. It doesn't stop at any one of those points. We get to a point where, can we manage this risk to an acceptable level that means that this technology, whatever it is acceptable? And that's the pathway to harm, thinking about how we define this in terms of environmental risk assessment. It's risk hypothesis based, and it's a conditional probability. This is about the only equation I dare put in this talk, but it tells me to tell you that actually, we're basing our risk evaluation on existing data and an existing hazard that we care about, and it's not independent of prior belief. And prior belief is really important. So that could come in the form of a qualitative assessment, or it could come in the form of a quantitative assessment. But that prior belief needs to be factored into when we think about when we're thinking about risk assessment, and that's really important. So let me just give you an example of where that might be important. So we identify a hazard. This is hypothetical, and it's just made up. And just to illustrate the point, that alterations to the population dynamics of an insect has a detrimental effect on wild biodiversity. We care about that from an ecosystem service perspective. The ecological implications of this, then we think, well, that's we've identified the hazard. We want to think about, well, what are the sort of exposure characteristics of that, and that involves thinking about altering some ecological process. In this case, the feedback that operates on the population. This idea of density dependence or overcompensation or competition for resources, whatever you want to think about that. So that's where we need our science to be able to sort of like codify this exposure. And then we ask the sort of hypothesis: is this density dependent regulation disrupted by the release of a modified insect? We define in a likelihood. That's a statistical framework in which to evaluate is the data that we have described by the hazard, and it requires us to have some model. That doesn't have to be a mathematical model, it could be a verbal model, and it requires some data to challenge that model against, and to say, is this the right descriptor for the data at hand? And we need this framework, or a framework like this, in order to be able to do sensible risk assessment. So I've set the scene. I want to get into now thinking about how we think about harm, because that's really important when we're thinking about regulatory, the risks of releasing GM insects. So this is a schematic which looks at the reduction in population size against comparators. And in the conventional plant world, when you're thinking about modifying your favorite plant, the sort of standard mantra is that you would compare that modified plant, to a conventional counterpart. So something that doesn't necessarily have the Bt toxin inserted into it, and you could look at do the risk evaluation on that. And in the EU, that's how our legislative framework is essentially set out. We have a comparator to a conventional counterpart, but that might not be that comparator approach might not be sensible for GM insects, or for the way in which we think about gene drive insects. So we might think of a whole suite of different comparators, and I just illustrate that in this schematic where, well, this is what I care about. This is my sort of in my case, and the way I think about this, the ecological drivers is in thinking about, well, the reduction in population size, because I'm caring whether this is an efficacious technology that's reducing this pest problem. Here's a bit of science that's shown me the GM has reduced the population to this level. Here's what happens in the absence of any control. And here are three alternative comparators. They could be different technologies. They could be the use of pesticides, or they could be something else. They could be actually, we might take comparators in terms of, well, if it's an insect vector of disease, is the comparator, is it some form of drug-based intervention? We might make a comparison there. Obviously, we might have something different on this axis, but it's really important to have these sorts of comparators in place. And what we're trying to do is what it says up here. We're trying to minimize this uncertainty in these adverse effects of releasing a particular technology or using a particular technology through better knowledge. We can't, just a bit tongue in cheek, we're going to call this science essentially. So there's a lot of science that underpins GM and a lot of science that underpins gene drive. I'm not going to go into this, but I just want to illustrate to you that actually we don't start from a blank sheet when we think about this. When we're thinking about gene drives, there's a whole set of research stretching back over almost a century on gene drive work, on selfish genetic elements that we can draw on to think about the regulatory frameworks that we build out. And here's just some examples. And again, I don't want to go into them. I'm going to pick on one. This one is particularly interesting because it highlights a whole set of things that I think are crucial to the way we think about gene drive regs. And this is the P element in Drosophila. What we can do, what these selfish genetic elements are doing is they're increasing their own frequency through a population when they would naturally, under standard Mendelian inheritance, be excluded. So we've got the frequency of the recessive allele, the one that would disappear if it wasn't under this drive selection, which is given by this very flat line here. And we know that knowing how in the heterozygous state this drive is operating tells us how fast that was spread through the population. So we've got some, we get, even from our old population genetics work, we were able to draw on this science to actually help us understand what might be classed as an adverse effect of these technologies. So our maths helps us here. Also, data helps us here as well. The P element in Drosophila is a transposable element. It sort of sits in chromosomes and leaps, and it replicates itself within the Drosophila chromosome, and it can increase its frequency within the Drosophila chromosome. So it's not a fixed drive. So it is something that occurs naturally. And here's just the data that was collected on P element in Drosophila melanogaster. So through the middle part, so like into the 1960s of last century, there were surveys of Drosophila melanogaster populations which showed that there was no evidence, there was no indication that this P element was in any of these naturally occurring melanogaster populations. By the 1980s and going forward, all naturally occurring Drosophila melanogaster populations have this P element in there. And that obviously raises a concern. It raises the concern that these things can spread very fast. It also raises the concern that the temporal and spatial extent of selfish elements from what we've learned from a natural system should be translated into the way that we think about gene drive regs. And that's probably the biggest thing. And I'm going to sort of give you the punch point of my talk right now. That is it. It's essentially that actually gene drives have the capacity for greater spatial and temporal extent than we may have seen in other controls. But I'll get into that shortly. We can also see with this P element that actually it has consequences for the individual performance of the Drosophila. So on the left, on this graph, this is their viability, their survival and fecundity, reproductive capacity on the y-axis against the number of P elements that are in these chromosomes of Drosophila. This is work that was done by Ains way back in the dawn of time. And what it shows basically is that as you get more of these things, they have some fitness consequences, has some costs to the Drosophila. Again, we can draw on this science to help us think about adverse effects. So we shouldn't neglect any of this, fundamental research that we do in thinking about informing our policies. Let me give you another example where actually understanding the ecology of a gene GM system really matters for understanding and minimizing these adverse effects. So here's a model. This is about as complicated as it's going to get. On the left-hand side, I'm going to look at the changes in the vectors. We're thinking about mosquitoes in the group. We're going to do GM release. It's going to affect the births of the vector. It's a self-limiting, it's going to be a self-limiting construct that's going to aim to suppress the mosquito population. And there's some competition amongst those mosquitoes for resources. So what we're going to do, what we can look at is how does changing the release rate of these mosquitoes affect the dynamics of the system. And what this sort of little movie is showing is what happens as I increase the frequency or the proportion of these modified insects I'm releasing to the environment. And what we can see, what we can see is that there are two states in the system. There's one where the system is at a stable state. That's what where the vector is now. And there's an unstable state where it's not in the system, but it could invade. And these are ecological conditions under which we have two stable states. As we start to increase our release ratio of these modified vectors, it changes the whole state of the system. It's changing the landscape. It's not just the parameters, but this is measuring the potential, the propensity of the system to change. When it's at a stable state, there's very little things that can make the system change. You've really got to change and what I mean by landscape, I don't mean that in the geographic sense, I mean that in the dynamic sense, that actually what it's doing is it's changing where in parameter space, populations can exist. I've got a stable point here. As I increase the release ratio, it collapses the system, and there's greater propensity for the system to change. And that's really important when we're thinking about, when we're doing our releases, when we're thinking about understanding what these potential adverse effects might be. Modeling is going to be crucially important because it tells us where systems might change, how much we have to push a system to make it change, and what the outcome might be. And the outcome in this case is that beyond a certain release ratio, when it gets up to about 2, we can collapse the system where the only stable state we see is the exclusion of the vector. Okay, so we've characterized this idea of the risk of adverse effects. I want to know harm 2 is thinking, is the effect harmful? So what we can do now is we can take the same graph, I'm doing the same schematic I showed you earlier. We've got the reduction in population size against those different comparators. And now there's a dotted line that says, this is harmful, this is not harmful. And again, we're trying to minimize uncertainty with clearer definitions of harm. So actually something's harmful, something's not harmful, and this gets us into the realms of policy. As Hector outlined at the start, I've been involved in various bits of different work over the last few years with EFSA, with the House of Lords, with the WHO and FNIH, thinking about how we can draw in our science into constructing relevant frameworks for GM insects. And gene drive, and more recently some gene drive work. But there's a whole set of existing approaches that we can draw on and work with sort of colleagues in Switzerland and the Netherlands. We've recently produced a review looking at these sorts of how we can use these existing approaches and existing approaches to environmental risk assessment to help us inform about this harm, no harm situation, and thinking about what that might look like. And we think there's four. I think there's 4 bits of existing risk assessment frameworks that we can draw on. One in terms of thinking about biocontrol, using classic sterile insect technique, using incompatible insect technique, and using self-limiting GM insect releases. And I'm going to talk a little bit for a few minutes about each of those. So biological control, probably very familiar to everybody, is where you're doing, and it's an intervention method, well-established intervention method. You find a pest, you go off to its native range, you find a natural enemy, you do a whole set of evaluations, quality control evaluations to think about whether that particular natural enemy is going to suppress this pest population in its non-native range. And we can sort of divide biocontrol into classic, where you do a single release, it establishes and it sort of suppresses that pest population, or an inundative control, where you repeatedly introduce the control agent. It's intended to establish, so it's a control agent that is intending to establish in an area of introduction, a bit like all of our thinking about all of our GM GM insects. It's also going to have some spatial spread, and it's also going to, in a classic sense, it's also going to persist temporally in the extreme, in perpetuity, to control that particular pest of interest. The environmental risk assessments, the ERAs, tend to, for biological control, tend to focus on the non-target effects, basically. What's the specificity of the control agent? What's the specificity of that natural enemy in its native range? And what effects might it have on non-target effects in the native range? So there's quite a lot of work around this quality control of the biological control agents. Where biocontrol regs exist, they often require risk-benefit analysis, which is different to the legislative frameworks that we have in place for GM. And it's something that we think we can approach with GM, and I'll get to that at the end of my talk, where we can actually use our harm frameworks to think about a risk-benefit analysis, even within existing regulatory frameworks that we have in place for GM insects. And there's obviously, this is within new EPO standards, there is now a requirement for having regulations that take into account risk-benefit analysis for biocontrol. So we've got quite a good, again, over the last 100 years or so, lots of good science evidence about how good and bad and how it works and doesn't work, how biocontrol is successful, where it's not successful. We're able to draw on that rich literature and understanding about how to inform our gene-derived regs. And really important is that you're putting a whole organism, like a new natural enemy, into a new environment. So that should sort of like give us pretty good understanding of the importance of this sort of control approach for developing consistent and adequate regs for gene drives. Sterile insect technique. So obviously, so like there is a GM version of this, but I'm talking about the sort of like classic approach to using the sterile insect technique. Again, it's an inundative, repetitive release of mass-produced sterile insects, again, directed at a particular target organism, first developed, again, in the 1940s, used in anger in the 1950s, a great screwworm in the Caribbean, very effective. The idea is that you mass rear your favorite pest, you blast them with some irradiation, you make them sterile, you release them out into the environment, and you have, and you expect that actually when they mate, they produce a non-viable offspring, or and then the population will decline. The sort of like the effectiveness of sterile insect approaches is proportionate, is sort of correlated with the fitness of those released insects. So the more you irradiate them, the greater the risk of having somatic mutations on that affect longevity, dispersal, sort of life history characteristics of the sterile insects that makes them less fit to perform against wild type insects in the environment. It's interesting from a regulatory perspective that there's that up until quite recently, so when I've been quite recently, up until about 15 years ago, the regulatory oversight for SIT wasn't necessarily well constructed. But in the last few years, it's now under some international phytosanitary standards. They're classed as beneficials, and there are emerging regulatory approaches for this. But it's still the regulatory, although the regulatory structures around SIT aren't well developed. Obviously, there's lots again of good science information that we can draw on. Again, there's without regulatory oversight or regulatory structures in place, it means that formal ERAs aren't necessarily undertaken, but where they are, they tend to focus around the efficacy of the technology. So things to do with the preparation of the sterile insects, but and also the sort of biosafety implications in terms of consequences for altering ecosystems, altering species, what the consequences are for collapse, or not collapsing, I mean, knocking species out of food webs, and the consequences of species replacements. So that's the sterile insect technique. Incompatible insect technique, so like thinking about the use of Wolbachia, so it's a bacteria. It's also a selfish genetic element. So again, we can draw on all of the rich literature and all the rich science that's out there on Wolbachia. It's disrupts, it's a bacteria that disrupts or biases insect reproduction in favor of its own spread. It was used, it has been used, it's been used against Aedes it was used in the 1960s, and more recently has been used for Aedes mosquito control. In the US, the regulatory oversight for the use of Wolbachia has been through the EPA, and it's been approved. So Wolbachia infected albopictus has been approved in the US. It's also been approved in Australia, but through some very convoluted routes, and eventually was classified or characterized as a veterinary chemical product. And again, there's good information and good science out there that tells us how useful this sort of technology could be in suppressing insect populations and or altering their refractiveness or their capacity to carry pathogens. So lots of really good science out there that we can draw on with this particular technology. The ERAs, there have been ERAs on for the use of Wolbachia, particularly in Australia, and really thinking about the consequences for horizontal transfer of the bacteria, which can happen and has been documented to happen, but also again thinking about what the consequences are for ecological food web effects. So again, one of the things that those ERAs conclude, which again becomes super important because it's a selfish genetic element, that actually monitoring, which is mandated under lots of legislation for GM, is now becomes a sort of integral part of the ERA and isn't something that sits after the fact of doing a risk assessment. It becomes part of that, and that's sort of advocated within the use and the regulatory frameworks that are being developed for the use of this incompatible insect. And then there's obviously we have a little bit of emerging, but not necessarily commercial, lots of field-based studies on the use of self-limiting GM insects. So these are similar to the SIT approach in their operation, in that they're aiming to suppress target populations. It involves repetitive mass release of a modified insect. They've been developed for mosquitoes in particular, and a bunch of agricultural pests. So obviously with lots of GM legislation around jurisdictions across the planet, they are regulated through that. FNIH, WHO have been at the forefront, have been developing guidance for GM insects and suggesting a phased approach. I'll introduce that in a moment. DRAs, so really most recently looking at doing releases in Florida and the use in Sabah, so in the US and in Borneo, so in Sabah through the Dutch agencies, really thinking about how to use our existing ERA frameworks for GM organisms, and really thinking about how they have been used, how they can be used to think about the use of GM insects. So we're stepping towards a regulatory framework that's now being more applicable to the sorts of things that we're thinking about here in terms of gene drives. So again, those ERAs are focused on all the things that are in GM legislation to do with horizontal gene transfer, effects on non-targets, ecological effects on food webs, alterations in fitness of the modified organism. And again, monitoring has been advocated to ensure efficacy as well as biosafety, to ensure that they're doing what they should be doing, rather than, as well as the implications for the wider impacts on the environment, human health, wider biodiversity. So here's just, so in this review that we put together, we can just highlight sort of the pathways, so like taking that pathways to harm approach that I talked about, and really thinking about how we can use that under these different technologies. And what this highlights, a complicated picture, I'm not going to spend a long time on it, but at the bottom we care about an ecosystem service like biological control. That's the one that we picked on. So if we've identified something that we care about in ecosystems that we don't want to lose because it provides us with an ecosystem service. So if we care about that, then we care about the reduction in the loss of natural enemies, and each of these controls give rise to various points of data. So the little boxes highlight things that we would need to know under these different regulatory frameworks to provide us with the right information to evaluate whether we want to do a GM insect or whether we want to do a classic biological control. So we can use this sort of approach to, it advocates the use of a pathways to harm approach and a risk hypothesis approach to identifying what the critical bits of data are that we need in order to develop a policy. So in the last few minutes, and I will stop soon, I just want to finish up. We're talking about, is this effect harmful, and think about the applications to gene drives. So we really want to think about, as I said, evaluating risk and evaluating an ERA for evaluating risk, and also mitigating that risk through management. And that has to be super important. That actually really thinking about the way that we can define harm, contextualizing what the comparators are, really understanding what that is really critical as this technology of gene drives moves forward, and having clearly defined pathways to harm that are risk predicated, not science predicated or belief predicated. And then that in order to get to that, I would argue, and I'll get on the soapbox, that obviously math modeling is super important, and monitoring from the get-go it has to be more central in these regulatory frameworks. And we have to think about these being logically consistent and proportionate to the technologies for use. So again, let me just illustrate that with a hypothetical for the release of Drosophila suzukii. So this is gene drives have been developed for this Drosophila. It's a pest of soft fruits. And again, down here, we care about the reduction in a particular ecosystem service by a control. So we can identify that a number of steps through this pathways where gene drive Suzuki could impact the capacity for biological control agents to persist in an environment. So it may be that actually the suppression of Suzuki means there's lack of food for natural enemies, reduction in natural enemy populations, and we lose our biological control. It may be that, and again, it requires the science that actually the gene Drosophila hybridize quite well, so that's a bit of science we know. Is that important for Suzuki? We don't know. We need some science there, and we can identify pathways through this where we need more data to be able to inform us about changes in ecosystem services other ecosystem services that we may value. So these again, I advocate this as a position in terms of the way we should construct our pathways to harm. And then this approach we had in the WHO piece of work a few years ago was having this phased approach to the implementation of a GM insect. So it sort of centers around efficacy and biosafety, and you can step through doing lab studies all the way out to open field releases. If you've got something that's self-limiting, and it will fade out over time. But gene drives won't do that, and so while this bit of the work may be okay, once you get out into once you're getting out to field trials, then you really need to think differently than we have done before about the regulatory approaches and the science approaches to this. And it draws on what I said, that actually gene drives are going to have greater spatial and temporal extent than we may have experienced with any other GM. But it doesn't mean to say that we haven't got a whole set of science behind us that we can use, that we can bring to bear, and a whole set of regulatory approaches that we can bring to bear on our understanding of how we develop appropriate regulatory frameworks for gene drive organisms. So I just want to sort of like, again, just illustrate very quickly some of the science that we can do with this, and actually an understanding how we might use mathematical modeling to help us understand the implications of GM releases for gene drive organisms. So we're looking at two mosquitoes, again, a bit hypothetical in the sense of it's a gene drive, but we've got Aedes aegypti, Aedes albopictus, which will compete. We're going to do we're going to do a GM release, a self-limiting GM release that's going to impact both the competition and the competition within and between those two mosquitoes. And we might expect, under classic ecological thinking, there will be a coexistence point in this system where those two mosquitoes might happily coexist. They may not, but we can draw on lots of science for us to think about that. What we can do is that we can evaluate using these mathematical models tells us what the consequences are as we do our release. So here we've got a system where the system is stable. It was stable here. I start to do my release and the system collapses down to the only stable point is when this primary vector has gone extinct. It ends up here. So the system is the landscape again, the potential for where we might see outcomes changes as we do our release. And it's really important that we appreciate that as a fundamental in when we do these sorts of controls. We're not just collapsing the population, we're actually, the parameter, in this case, the release ratio, is actually altering the potential for coexistence. It's changing the shape where this coexistence can happen, and it's leading to a point which is the outcome of exclusion of the primary vector. We can see the same situation with a different state, with a system now where we don't see the system in a stable state. There's two mosquitoes either live independently, and what happens here is that as we do the releases, we erode that stable state for the vector we're trying to control, and we extend out the space over where this secondary vector lives. So I've talked about that actually we can change the space over which this species lives in and alter its sort of like its niche breadth in that way. What this leads to is an alteration in the epidemiology. So we can think of which again might be a comparator that we care about with vector-borne diseases. It might not we might not care what the implications are in terms of the ecosystem services. We might care about what it means in terms of human health. So again, we can build a mathematical model that allows us to evaluate, the outcomes of these different controls, these different ecological outcomes of coexistence or exclusion and its epidemiological consequences. So here we have the situation where those mosquitoes were initially coexisted, and we do a particular GM release, and this is the proportion of the vectors of the primary vector that are infected, secondary vector that are infected, and then the distribution of infections across hosts. But what I want to show is that the way in which the ecological system set up, coexistence versus exclusion, having a stable point versus two unstable, points where those vectors weren't necessarily interacting, can give rise to completely different implications for the epidemiology that we see. And that might be really important when we want to construct out an understanding of whether the release is harmful in any way. So I'm about to finish. Sorry, Hector, I'm about to finish. Don't worry. Is the harmful effect acceptable? Well, it's only so. What we can do now is we can take this graph again. I've shown you this twice. This is the third time. We've got our comparators. We've got a situation where we separate out harm from not harm. We've now have a situation in which we add a third tier, which is actually that harmful effect is acceptable or unacceptable. And it may be that this difference between what we're trying to do and what might be no control or other comparators gives us a measure of the benefit of doing this technology over doing nothing versus the others. So with this approach of being able to characterize harm, we can identify a way in which to evaluate the outcome of a technology without necessarily calling it benefit, but actually evaluating this in the right comparative way to give us an understanding of the costs and benefits of doing one technology, using one technology for control over another. And in order to do this, we need, again, tongue in cheek, we need more leadership. Okay, so what I've shown you is that this pathways to harm and the formal definition of risk is really important. We need science, we need policy, we need leadership, we need good science, we need good, in my world, good ecology in thinking about this. There are existing science and regulatory frameworks for us to use. And I'll leave you with the thought that actually any policy that we develop for gene drives or any policy we develop for any technology needs to be economically viable, proportionate, and logically consistent to what we're trying to achieve. Thank you.
Oh, I'm sorry. I just realized that I wasn't being heard. But oh, I was going to say I'd like to encourage people to encourage people to write down their questions and then we'll deal with them after the presentations are done. Can people tell me whether they can hear me?
Yep.
Yeah, I can hear you.
Okay. So with that, I'd like to turn things over to Owen. I apologize that I didn't have my mic on, but I'd like to now turn it over to Owen to talk about what can be brought to the table by invasive species. So Owen, it's all yours.
Thank you, Hector. Can you see my slides?
Yeah.
Yep.
Excellent.
We can see it. Yep.
So thanks for the invitation. I appreciate the opportunity to talk to you. And as Hector said, I work for CSIRO, which is the National Science Research Organization in Australia. And I work for something called the Synthetic Biology Future Science Platform, where I oversee, amongst other things, pretty much all of the genetic biocontrol work that goes on in my organization and across Australia. And that's not just the technical aspects of it. We do develop using our synthetic biology, but we also are consider all of the ecological and social factors. So we do a lot of ecology and population genetics and social science associated with it as well. I'm going to be talking specifically about invasive species risk assessments and their relevance towards the regulation of gene drive organisms. So I'm just going to give an example of a pest risk analysis that's available for invasive species, one that's put out by the IPPC. And then I'll talk a little bit about an annex or an appendix to this particular risk assessment protocol that talks about how it should be applied to living modified organisms, or GMOs, and since this makes it more relevant to gene drive organisms. And then I'm going to focus on a bit of science in that I'll make the argument that the most relevant risk is that's between invasive species risk assessments as they're relevant to gene drive organisms is the issue of spread after release. And I'll talk about some of the ways in which we have developed tools to measure rare dispersal events when it comes to invasive species, and how those can be used to assess movement of gene drive organisms. So this is the pest risk analysis that I'm using as an example. It's from the International Plant Protection Convention, supported by the FAO. It's officially the international standard for phytosanitary measures number 11. I'm sure that's of interest to absolutely everybody. But basically, it goes through the stages of pest risk assessments. It's very similar to the ones that Michael has described previously. Going through the stages, assessing the probability of introduction. In this case, it can be a species that's new to an area, or a genotype or phenotype, such as insecticide resistance, that's new to an area. Attempts to quantify the probability of establishment and spread. Assessments of potential economic and environmental consequences, and then assessments of how those consequences can potentially be managed. So in terms of the first stage, the probability of an introduction, one of the key factors is whether it's present or absent in the area that you're addressing. So in this case, whether you're talking about a new species to an area or a new genotype or phenotype to the area. But mostly it's focused on the probability of entry, and that's associated with entry pathways, which have to be identified. And then consider things like the probability of survival during the transport, the probability of surviving pest management processes that happen before, during, and after transport, the probability of locating a suitable environment upon entry. And so that a lot of times has to do with the phenology of the pest organism, whether it's an insect, a disease, or a weed. In terms of the next stage, the probability of establishment, there's assessments of the suitability of the environment, questions as to whether there are the presence of host vectors or other species that are necessary for the organism to survive in a particular environment. Like some of the assessments that Michael was talking about, a lot of emphasis on the characteristics of a pest, which affect its probability of establishment. And then again, this what I think is a really important one when relating this to gene drive organisms, the probability of spread after establishment. And I'll come back to that. Of course, there's the assessment of potential consequences. Most of these in these PRAs are generally focused on primarily on economic damage, and this can be direct or indirect. And by indirect, it can mean that's through community processes such as competition, predation, etc. It involves an assessment of pest characteristics often. But there's also assessments for the potential of environmental damage and the potential for social damage or social consequences. And this is just an example of a PRA. They are quite often developed with respect to a particular ecosystem or commodity. In this case, it's a PRA with a focus on Hawaiian forests, and then an assessment of all of the pests that can potentially be potentially may enter, in this case, Hawaii, and then assessments of the likelihood of introduction and the consequences of introduction. And you see each of the characteristics of each of those here. And then in this case, it's just a qualitative assessment, high, medium, or low for each of these species. And then this is summarized, and the species are generally ranked. In terms of the pest risk management stage, there's considerations for options for preventing or reducing infestations once they've occurred, options for maintaining pest-free regions, so keeping the pest out of particular areas, and then whether it's possible to set up regulatory barriers, such as prohibition or phytosanitary certificates to help contain. And these regulatory barriers are especially important for weeds because plants that become weeds are often come from horticulture, often from people's gardens. And so there's a big focus on whether there are regulatory barriers that can prevent that from happening. And this is just an example of a weed risk assessment. This was actually done by the Department of Agriculture here in Western Australia, and is used as a model for many risk assessments around the world, and with a very, very strong focus on undesirable traits. And so these are traits that you would hope would not find in plants, and which make them more the capacity increase the capacity for them to become weeds. I just wanted to mention briefly, as I said, that there's this annex to this this document which talks about the pest risk analysis for living modified organisms, or GMOs, and it's very useful to, in this document, it states clearly that it's very useful to consider risks relative to the unmodified organism. And so you're not trying to assess the risk, because quite often the organism may already be there, which would also be the case for gene drive organisms. It's not the risk of the organism as a whole, but just the risk of the modification that's been done to the organism. And again, we want to consider direct effects and indirect effects. The direct effects, in this case, altered pest characteristics, which Michael was referring to in his assessments that he went over. And then the indirect effects, which is the quality or prevalence of other species due to the effects of the modification. And this can be if you put in a toxin like the Bt, whether that toxin can have effects on other species within the environment, or whether just the reduction in the pest of the reduction in the in other species within the environment can have detrimental effects on ecosystem services. But as I said, I think one of the primary points where we can get lessons for assessing gene drive organism from this sort of a PRA is around this question of containment risks for GDOs. And in particular, the question of the probability of spread after establishment. And so what I want to spend the rest of my time doing is talking about the ways in which we are able to assess or quantify rare dispersal events. In particular for invasive species, we're looking at events of incursion. So we're looking for those rare events that bring an invasive species into a novel environment. But I will argue that the same tools can be used to assess the escape of GDOs from the target environment, potentially into other jurisdictions. And so one of the approaches that we use is with modeling. And this can be done focused on natural or wind-borne movement, in this case, insects or fungi or spores, or human-assisted movement of invasive species like insects, animals, and the seeds of plants. And human-assisted movement is really important because most invasive species are invasive because they're very good at hitchhiking in boats, airplanes, and cars to assist their movement around the world. So to focus first on natural movement of insects and other organisms, we actually developed at CSIRO a tool called TAPIS, Tool for Assessing Pest and Pathogen Aerial Spread. And this is a modeling tool that was using a model that was originally developed by NASA to predict the spread of nuclear fallout from nuclear bombs, still is used to model the spread of ash clouds from volcanoes. And we adapted the model put it into a finer scale, included biological data for the organisms that we were trying to model, and looked at predicted how they could be spread by wind. And so the biological parameters that go into that are parameters that affect whether they get into the wind stream, whether they survive while they're up there, and also how likely they will be deposited somewhere where they're likely to survive. And just as an example of the model here, on these 4 graphs images, I'm showing the northern part of Australia and then islands within the Indonesian archipelago. And we're asking specific questions about movement from one of those islands in Indonesia to the northern part of Australia or vice versa. And the top 2 graphs, we're looking at the wet season and the dry season, and you can see that the wind patterns change considerably between the 2 seasons. But what we do find is that the wind patterns are sufficient that we would expect to see, in this case, we're modeling insects, we would expect to see insects being blown from an Indonesian island onto the Australian continent during the wet season, but not during the dry season. We then ask in the bottom 2 graphs the reverse question as to whether an organism from the Australian continent is likely to be blown onto the island in Indonesia. And again, looking at the 2 seasons, in this case, the wind direction is appropriate in the dry season, but the winds are actually not strong enough that we would predict that you'd actually get deposition of insects into the Indonesian islands. And this approach was actually used to look at a fly, which was a vector of a livestock disease, blue tongue virus, which is prevalent in Indonesia and not prevalent in Australia. So assessing the risk that a fly would be, a vector would be, that carried this virus would be blown into Australia. And so basically there was, using the model, we were able to show that we would have an expectation that this would occur. Also very interested in modeling human-assisted movement of insects, animals, and seeds. And we do this by looking at this wonderful depiction here of the shipping lanes within our region. So this is the Southern Pacific, Indian Ocean, and the various seas around the archipelagos in Southeast Asia. And you see it's a very crowded shipping lane system. And just as an example, we can ask the question, what's the likelihood of entry of a particular pest? In this case, it's called the capra beetle, which is a pest that bores into wood, into Australia via shipping lanes. And this is a particularly good hitchhiker because it's basically found inside of wood and is hidden from detection. And the countries here in gray are the potential source populations and the respective ports. And so using the shipping lane models that I showed on the previous slide, we ask questions about any particular port's potential to be the source of an infestation into Australia and the potential of this beetle arriving at any particular port within Australia. I want to finish off by talking about the tools that we're now using for monitoring incursions, which are population genomic tools. And here we basically sequence the full genomes of individuals that arrive into the country and then compare them to full genome sequences from all the potential source populations. Now, the analysis in this case is agnostic to natural or human-assisted movement, but I'll show you in a second how sampling can be used to target particular movements such as human-assisted movement, so along particular pathways. So again, here we have this wonderful diagram of shipping lanes. And here we have we're interested, and this is work that was done in Ari Hoffman's lab at a collaborator of ours at University of Melbourne. And Ari was interested in Aedes aegypti and Aedes albopictus. Now, Aedes aegypti is present in Australia, but only in far north Queensland here. Aedes albopictus is not present within Australia. And so again, we have listed all of the ports, etc., where these species are present, which are the potential source populations. But for a particular analysis that they did, they did sampling at Australian airports to ask the question as to whether any of these Aedes mosquitoes from either species were being introduced into Australia via airplanes. And what they tested at 6 different airports around the country, none of which were endemic for Aedes aegypti, and obviously not for albopictus either, because we don't have it in Australia. And what they discovered is that the Aedes aegypti that were caught at all of these airports all came from 3 different locations in Southeast Asia, none from the endemic populations, and the Aedes albopictus could be traced back to 4 different airports, again within Southeast and Eastern Asia. We do similar analyses for pest insects. This is actually the Queensland fruit fly, or the Q fly, which is found in Australia and also on neighboring Pacific islands. And so we do full genome analyses like this, which and aim to separate the populations. The Pacific Island populations are, and some of the Australian populations are quite distinct. And if you actually look a bit closer, you can see that even some of the mainland populations of Australia are genetically distinct and can be separated. And the consequence of this is we're able to come up with 6 different potential source locations from which flies could be distinguished to a likelihood of greater than 90%. And this could then be applied to incursions, for instance, into New Zealand. And so we tested flies that were collected at Auckland Airport in 2014. In this case, there were 8 flies. And with 95+% certainty, we were able to say that those came from New Caledonia. And if you look at shipping lanes, this New Zealand, that's New Caledonia, there's actually quite a bit of activity between those 2 places. There's also a lot of activity, not surprisingly, between New Zealand and Australia, which explains why in 2018, some Q flies that were established in an Auckland suburb sorry, I've gone too far. In 2014, we had 8 Q flies breeding in suburban Auckland, and these could actually be traced back to the east coast of Australia as the source population. And no surprise here, in 2018, another 2 Q flies that were established in an Auckland suburb were traced back again to New Caledonia. So we, using these tools, then we're able to trace back rare dispersal events that result in incursions where the species don't already occur. I believe that prior to doing releases of gene-drive organisms, the same sorts of analyses could be done for the species that's already present in the area, in the target area, and ask the question as to how often those individuals from that population are moving to other countries as an indication, an estimation of whether a gene-drive organism might do that in the future. And in particular, we could do that around ports and airports to look at human-assisted movement, which in general is more common than natural movement. And the other advantage of gene-drive organisms is that in the monitoring period, we actually have a genetic marker that's present within those gene-drive organisms, and work that one of my colleagues has done in CSIRO, Dennis Bauer, who's a bioinformatician, has shown that in pooled sequences of large numbers, hundreds and thousands of eukaryotic genomes, it is possible using a K-mer analysis to very accurately detect whether any of those organisms were carrying a Cas enzyme. So in summary then, I've showed you that there's an IPCC that they formulated a risk assessment protocol for invasive pest species, that some of the latest protocols have included additional risks posed by living modified organisms, or protocols for assessing those risks, which makes them more relevant to gene-drive organisms. But in my opinion, at least, the risk of spread after establishment is perhaps the most relevant risk that's assessed as part of invasive species, relevant, that is, to gene-drive organisms, and that the modeling and genetic methods that we've used to quantify rare dispersal events for invasive species can be applied to gene-drive organisms, whether you're assessing natural or human-assisted movement. Thank you very much.
Thanks, Owen. I'm checking to see whether I can be heard.
Yeah.
Yes,
Yep.
you can now.
Okay, great. Thank you. So this is a time now for questions. There is one person who has raised their hand in the panelist in the participant box. This is Werner Schenkel. I'm going to turn on your mic, and you can ask the question that you have.
Hello, am I heard?
Yes, we can hear you.
I'm very sorry. This was just a technical mistake from my side. Sorry.
No problem. Oh, so you don't have a question?
No, I don't have a question. I'm sorry.
Okay. All right. Okay then. Then we'll go to the questions that have been submitted from on the chat. I think the first one goes to Mike Bonsall from Jim Lavery, who says, in legal theory, a harm is conceptualized as a setback to the interests of a specific stakeholder or stakeholder group. This conceptual link to the experiences and interests of individual stakeholders or stakeholder groups wasn't clear in the section on the acceptability of the potential harms, or you might expect it would have the most direct relevance. Could you address how this fundamental aspect of harm figures into the models or their application? Mike, I think that's over to you.
Yeah, great. I think thanks for the question, Jim. That's a really great question. So one of the ways, so yeah, I didn't get into that in the talk. Obviously, involving stakeholder engagement is really important in all of this, and thinking about how you then use that information in risk assessments is really important. I think one of the ways, I think the best way I've seen, one of the best risk assessments I've seen done was for Wolbachia, the use of Wolbachia in Australia, where they're using Bayesian belief approaches to doing this. So a statistical framework in which you can build in belief. And that belief can be quantitative or it can be qualitative, and you can assess the outcome in terms of whatever your ecosystems in this, whatever your hazard might be in that way. I think if you wanted to develop that, so if you wanted to incorporate that into a sort of more quantitative risk analysis framework, that would be your best bet.
Thanks for the answer, Mike. I'm going to switch over to a question for Owen. Owen, this is from Jason Delborn. How might we account for intentional human transport of gene-drive organisms motivated by their potential to impact a different population than where it was released.
Mm. Oh, thanks, Jason. um, yeah, I'm. I think we will. I think we're likely to struggle to be able to having had experience with this with rabbit biocontrols within Australia. I think it's going to be really hard, certainly within a country, except by policing it very closely. I think internationally, if you do have a secure border system like we have in Australia, it will be easier, especially being an island. In other circumstances, like contiguous countries on other continents, I can't actually think of a way of doing this. I think it would probably have to be addressed through communication strategies, emphasizing the risk of those approaches in terms of our capacity to continue to be able to do it in our own countries. But I'm afraid I don't have a strong answer to that, Jason.
Thanks, Owen. I'm gonna kick it back now again to Mike from Stephen Panossian. This is to Mike. How do you quantify exposure to risk as an observable in the field? Yeah, this is from the beginning of your presentation.
Yeah, great, thanks. Another great question. Thank you, Steve. Yeah, I can give you an analogy more than what I might do in the field. Which I often use is I cycle. I use a bike. I cycle a bike around Oxford, and my the hazard of cycling, a hazard is just being hit by a bus. But my exposure to being hit by a bus really depends on where I'm actually put my bike, either in the middle of the lane or in the bike lane or whether I'm wearing. So where I sit. So while the hazard is consistent, the exposure isn't. It really sits, or it really depends on where I am with my bike. And then my risk mitigation in that, and I think that's part of this, is that obviously I wear a fluorescent jacket and I'm wired up like a Christmas tree. So I think it's a really good question about how we sort of like quantify exposure to a risk in the field, and understanding from a gene drive perspective. It may be, for the example that I gave, thinking about Suzuki, as that where gene drives have been developed, you want to think, well, what is the sort of like the hazard might be the potential for the potential for hybridization. The exposure might be, well, do you have species which Suzuki will hybridize with in that locality? So I think it's really predicated on us having a richer understanding of the system in which we're doing those releases to. So I hope that gets to some point of answering the question. But I think being able to quantify those things is critical in thinking about how we evaluate risk. They're not easy, and finding pragmatic ways to do that is really important.
Thanks, Mike. This next question is from David Obrakta, initially for Mike, but I think I'd like also to ask Owen to address this question too. David asks, while the parallels you've drawn between GM insect technologies for pest suppression and say biological control, or in Owen's case, invasive species seems valuable, these seem to be completely absent from the public discussion of these technologies. Why do you think that is, and how can these broader perspectives be injected into the discussions? Mike, will go with you first, and then Owen.
Yeah, good question. Another good question. Great sets of questions. Yeah, why has it not been? So yeah, those technologies. I guess we lived in a different world. I think is the answer to that. Is a straightforward answer to that. Maybe we don't now. It's really important that we are able to use this, use these existing frameworks and our existing science to inform on gene drive regs. I think it's understanding that we're not starting from a base. We're not starting from a where we don't have any evidence. We do have all of this stuff. That the point of the the review we've put together is really to highlight these sorts of where we can draw on existing approaches to really think about this. And I think that's quite novel. I've not seen that done before. But injecting these conversations like this webinar is just a good opportunity to get to see what people think about that, how we should be engaging on a broader dialogue about the use of incompatible insect technique or sterile insect technique, or more importantly, biocontrol, which since in many ways has sat below the radar of any of this stuff. I guess with the GM stuff and the incompatible insect approaches, there's a growing momentum behind that for sort of like engaging stakeholder groups through these sorts of dialogues. So yeah, it's just like that. The thing that Gene Conveners is doing and is built on is a platform for us to be able to to have these sorts of conversations. So yeah, I guess it's watch this space in that sense, right?
Yeah. Owen, how about you? Owen, can you hear us?
Sorry, I was giving a wonderful explanation, completely muted. Now I've forgotten it. I think part of the problem is that there's just so much of an emphasis on genetic modification that immediately people's are immediately wanting us to regulate the fact that something is genetically modified as a priority, as opposed to assessing the other potential risks associated with basically the change to the biology that you've done to the organism. I will say that the risk assessment that was done for the Wolbachia population replacement work is a really good model for doing population replacement work with a gene drive organism. And whether it's correct or not, of course, the Wolbachia was not considered to be genetically modified with the insertion of the Wolbachia bacterium. But because it avoided the path of regulation that had to do with genetic modification, I think it actually slotted in to a different regulatory path, which resulted in a much more efficient and a much more relevant risk assessment. Whereas the problem with slotting it into the path that goes along genetic modification is that most of the regulatory pathways for GM have been designed for GM crops and have not been modified for most other applications of GM. And so if you do take it along that path, you end up asking a lot of questions that aren't relevant and avoiding a lot of questions that are. And so I think we can. I think something needs to be done to make sure that gene drive organisms are actually considered under the most appropriate regulatory framework and not just immediately slotted into a pathway for GM.
Yeah, what you're saying there, Owen, is kind of interesting. I think this is kind of the direction I see Australia going in terms of the regulatory approach, where they're taking a more inclusive view of the biology of the organism and their pest risk characteristics as opposed to the fact that they're just GM.
Yeah, unfortunately, the way that it works within Australia is that ours, because it's GM, a gene drive organism will be primarily, I mean, the lead organization, the lead regulatory body will be our GM regulatory body, but under our legislation, they're required, because it's a population control mechanism, to bring in other regulatory bodies within our system and basically partner with them to have them assessed using the other regulatory framework that we have. So that's theoretically, I think, the path that would be most successful if it worked as it's supposed to work within Australia. But obviously, every jurisdiction is different.
Thanks, Owen. I've got a question that I'm going to go back to Michael on. This is from Teresa Njoroge. Please elaborate on the difference between ecologically confined field trials and physically confined field trials in the context of risk assessment.
Yeah. Okay. So I think physically. So when you're thinking about, there's been lots of conversations about this around how you might take that phased approach to understanding the environmental risk, doing an environmental risk assessment for gene drive organisms. And one of them is to think about doing it in a physically confined space. So, I put this in inverted commas, finding an island somewhere where you could release the thing where it would go nowhere, but except across that island. So that will be a physically confined space. Okay, but that might not be that'll only give you part of an information about your environmental risk assessment, because you're not putting into an environment where it could where you might have the necessary, it might not represent the places where you might want to do that control. Ecologically confinement means that actually it's confined in a space where it has the propensity to spread, but because of ecological conditions, it's not going to go. It's not something to do with the local environment prevents it from going. So again, you're only going to get part of the information for your risk assessment. So I think one of the things here is that phased approach might not necessarily be the best regulatory pathway for understanding gene drive organisms. And as I said in the talk, I think that one of the things is really important is that we consider how to, not necessarily it's not necessarily the field trial per se, but it's how you develop monitoring schemes and assessment processes for the releases we do from the get-go. So and again we've talked about gene drives in a general way, but there's lots of, once you lift the lid of that, the technological developments that are in place for gene drives, and I'm sure they've been discussed at these webinars previously, the science behind that is growing daily and is very rich. And that what we might have classed as a global drive, like the Drosophila PM element, which you may want or you may not want, but developing those technologies so that actually they become spatially confined and temporally confined because of the technology, because of the way in which they work molecularly, is much richer and allows us to actually think about doing gene drive regulations in a much more case-by-case way than just thinking about blanket regulations. So while that might become administratively burdensome, doing them on case by case, it means that we're able then to think, well, actually, if you've got a split drive that's trying to control Suzuki here, you're trying to use a global drive to do Melanogaster here, you've got to think about how those regulatory frameworks grow out in different ways, and beyond the question to do whether they're ecologically or physically confined. And it comes down to thinking about doing monitoring from the get-go.
Thanks, Mike.
Thanks.
There's one last question that I'll stick with you on. This is from Jason Delborn again. How does your call for leadership in your talk interact with the calls for public engagement and community input on decisions about gene drive?
Yeah, that's a great question. I mean, I think it goes back to sort of thinking about, the sort of things I was talking about with in answering Jim's question. I think it's really important that we are able to have these stakeholder and community engagement input into this, and we listen properly, and we don't actually. There are two sides to all these coins, and we need to listen to them. So, for some of the work that we did, some of the work we've been involved with EFSA, we held a public open forum where we could have those stakeholder engagements. And you might think that some of those conversations were frustrating, but actually it was really good to be able to hear what the concerns about the technologies were, to actually think about how we approach them, how we understand them, how we adapt them into our thinking about developing regulatory frameworks and thinking about this. So I think there's that call, if it is a call for leadership, and it was a bit of tongue in cheek, is actually that we do more of that. And again, I think this environment that the FNIH and the Gene Convene teams are putting together is just a fantastic opportunity to do that. It's an opportunity where we have this enterprise to be able to have these conversations, and they're constructive conversations. At the end of the day, there's no point throwing toys out of cots at people about this, or at people about this. It's actually understanding the perspectives in the right sort of way to get a fuller, richer understanding of these technologies in the 21st century, which, going back to David's question, was different than maybe have done in the 19th or 20th century. We're in a different world. Thank you.
Thanks, Mike. And a disclaimer, Mike is not paid for his endorsement of Gene Convene. Well, I think that brings us to the end of our session. And thank you so much to both speakers for being available to us, especially Mike Owen, who is well past midnight now in Australia. So thank you so much for that. And hopefully that makes up for your staying up late. But stay with us then next week. 2 weeks from today, for the next series, next couple of talks in this series on the regulatory and governance challenges posed by gene drives. Again, this is going to be 2 weeks from today, November 18, 2020. And there we'll be having Natalie Koffler from Editing Nature and Jesse Reynolds, UCLA School of Law, speaking on this topic. Look forward to seeing you then, and thanks again for joining us. Thank you. Goodbye, everybody.
Thanks, Hector. Thanks. Bye-bye.